The electromagnetic spectrum, longer wavelengths here, less energy, higher wavelengths up here. Again, frequency and energy or strength of the wave coincide, right? As the frequency increases, so does the energy in the wave. Wavelength, as wavelength decreases, so wavelength and frequency have an inverted relationship with each other, right? As wavelength increases, frequency decreases. And remember, frequency equals strength. So again, wavelength also has an inverted relationship with the strength of the wave. Cool? And again, all of this leaves the sun at the same rate, speed of light, blah, 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 right? All we get to see is in between 420 and 700 nanometers of that light, of that energy. That is all we can see with our eyes. Cool? So again, it travels in waves. Wavelength is the distance from the crest of one wave to the crest of the next. We drew that on the board Thursday, right? Not all of this energy here makes it into Earth. A lot of this up here gets blocked by our ionosphere. The magnetic sphere around the Earth and the ionosphere block the harmful rays. This is why, walking around on Earth, you don't need a spacesuit. Going out into space, you do need a spacesuit because you're being bombarded with all that radiation from the sun, right? Every bit of this radiation is hitting you. So the damaging rays are going to hit you, right? This is why they wear that. Also, if you didn't have a spacesuit, you would burn on one half of your body and the other half of your body would freeze all at the same time. The side not facing the sun would freeze solid, the part facing the sun would just get burnt to a crisp because of this radiation. Right, so when they go out into space, all this, right, stuff has to happen. We good? Entire range is known as the electromagnetic spectrum. Sure, we went over that last week also, correct? If you took physics, you should know this. If you haven't taken physics yet and want to take physics or are going to take physics at some point, anywhere there is an electrical field, there is also a magnetic field. And everywhere there's a magnetic field, we've got our little electrical field. This is why we call it the electromagnetic spectrum. Gotta let my shot. You remember the right-hand rule and all that for the way currents are going? Again, like I said, okay, so I said 423, 80 to 750 nanometers. That's the visible light. That's all we can detect. And remember, nanometers, tiny, 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 right? Nanometer. And that number is the wavelength. So that's what, when we say the nanometers and the meters and all this, that's the wavelength. So the bigger the number, the bigger the wavelength. The bigger the wavelength, the less energy and less frequency. We good? Everybody okay? Moving up. What? You go back. Okay. Okay. All right, so understanding light. Again, this is just background information. Don't get too wrapped up in this stuff here. What we're going to next is going to be more important. But so this is exactly what I said on the last slide. Short wavelength, higher energy. Long wavelength, lower energy. So if you look at it, here you have violet, here you have red. 700 nanometers, 400 nanometers. Which one has more energy, the red or the violet? Which wavelength has more energy, red or violet?
Red. Violet.
It's violet. Short wavelength, higher energy. 400 nanometer wavelength, 700 nanometer wavelength. Right? Shorter wavelength, more energy. So violet, a lot more energy than red. Cool? So when you look at the Roy G. Biv, red is the weakest of the visible lights, violet is the strongest, and it goes in order. Right? Of Roy G. Biv. Everyone knows what Roy G. Biv is, right? I hated it first time I was in a class and someone was like, and the teacher was like, Roy G. Biv, Roy G. Biv. And I'm like, what the hell is Roy G. Biv? And then somebody told me, I was like, oh, duh. But
Wait, Roy G. Biv, red, orange.
yellow, green, blue, indigo, violet.
Oh.
Everyone forgets indigo.
What's
Not the Indigo Girls. That's a band from the 80s or 90s. Indigo Girls. You guys ever listen to Indigo? Look that up. You're welcome.
Absolutely not.
When light interacts with matter, it can be reflected, transmitted, absorbed. So it's either reflected, bounces back out, transmitted, going through, or absorbed. The three things. We're not worried about refracting like that. What? We're doing reflection, not refraction. Refraction is fleeting. So this white light here. So the light from the sun is what's interesting. This light right here. Everybody knows if I put a prism up at this white light, what's going to happen? It's not going to be reflected. It's not going to be well, it is in a way, yes. But it's going to be split into multiple colors. White light, this is what we're saying, white light is all the colors that you can perceive. Right? If I put a prism up to a white light, what am I going to get out the other side of the prism? You guys never looked at anything from a prism? Okay, well, so you get the Roy G. Biv, right? It comes out in that order. Right? What's a rainbow? Why do we have rainbows after rain? It's not reflected. Well, that's where it's coming from, the sun, yes, absolutely.
Oh, the sun is coming down onto the water prisms.
Sure, the little waters, the water in the atmosphere or whatnot, is creating like a giant prism effect. That light from the sun hits that and is refracted. When we say refracted, what we mean is the light is split. It's still being transmitted, but it's refracted, bam, split into the colors of the rainbow. Right? That Roy G. Biv, this is what's happening. It's still being transmitted, but it separates it by wavelength. Which is just, it's an amazing phenomenon, right? Has nothing to do with leprechauns and gold. Okay. Pigments are able to absorb visible light. The color we see is the reflected wavelength. This is why when we talk about light and stuff, I tell kids, like, look, this right here is every single color in the world except for red. This hoodie is every single color in the world except for that specific wavelength of blue. There are different wavelengths of blue. If you look at this, there are different shades of blue. This is where we get all our pigments from, right? That color blue, this color pinkish something or another, that's in here, right? All of that is in there. What happens is pigments can absorb light. All of those things get absorbed. All those other wavelengths get absorbed. This exact pigment right here cannot absorb that wavelength of light. So it's reflected back out, and that's what you see. This leads right into this. Why are plants green? are the plant leaves green?
Oh. Is this the pigment the sun gives you?
The sun doesn't give pigment. You see them, right? What does that tell you about the light? If you see it, the light had to have been reflected out. If it's reflected out, what does that mean about the pigment? The pigment is green, that cannot absorb whatever green that is that you see, it can't absorb that wavelength of light. It's absorbing all the others. It can't absorb that one. Are all plants the same color green? No. Right? They absorb different wavelengths. Some plants can grow in some areas and some can't, right? This will have to do with partly the wavelengths that they get predominantly in that area, right? This stuff goes deep. When everyone says bio is so easy, man, it is. You can go so incredibly deep with this stuff that it's like nuts. There's so many fields you can get into. So many things you can explore. It's so fun. All right. Leaves are green because chlorophyll absorbs violet, blue, and red light, so it reflects green. Well, Mr. White, you didn't mention orange or yellow. We'll get to orange and yellow. What about leaves that change color? Yeah, so it's reflecting the green light, it's absorbing all the others. Now, in here, energy, right? This violet, blue, the red, it's taking those in, but there's energy associated with each of them. Not all energy is good energy. Well, all energy is good. Some of it is not good for the plants. Are we good? Moving on. All right, photosynthetic pigments. I know yours is not in color. But here, right, absorption coefficient, how much is being absorbed, right? Wavelength, 300 to 800. And chlorophyll A and chlorophyll B. Two types of chlorophyll that we're going to talk about. Chlorophyll A, if you look, it's right here, it's absorbing light, absorbing a little bit. Ooh, it's absorbing a lot right at the 420 nanometer area, and then it drops off. It is not absorbing any of this, and then it absorbs again. Where is it absorbing? Right in here, near the 700. Right? That's what this is telling us. Chlorophyll B, no absorption here. Ooh, it's absorbing up here. Again, no absorption. And then it absorbs up here in the 650 to 670, and then no absorption again. That's what this is telling us. The absorption spectrum. Telling us where in the spectrum of light it is being absorbed. So chlorophyll A, primary pigment in the thylakoids. Involved in light-dependent reactions. It's our blue-green pigment, right here. Chlorophyll B is an accessory pigment, meaning, so like when you accessorize, you add to things, right? It's something extra. This is our accessory pigment. It's not the main pigment for photosynthesis. the yellow-green pigment. Again, this isn't too deep right now. Do we have option output? No. And what they did to figure this out is they could take chlorophyll and centrifuge it, break it up and stuff. They put it in like a test tube, right? And they shine a light, and we have our board over here, and we see what wavelengths still come through, right? And this is how they figured that out. What wavelengths of light are transmitted through, which are absorbed, and which are kicked back. So this is kind of what they did to figure out the wavelengths that photosynthesis is using, right? I can't remember what this is called, dude. It's a spectrometer of sorts. I can't remember which one it's called. Sorry. Carotenoids. These are the goofy ones. Well, not goofy ones, but these absorb harmful rays. It's for photoprotection for the leaf. So broaden the spectrum of colors that drive photosynthesis. Yellow, orange pigment. They absorb and dissipate excessive light energy that could damage chlorophyll or interact with oxygen. So this is what protects the leaf from what would be harmful or unusable light energy. So we don't see the carotenoids on that last chart. Who's that? Yes. Yeah. Are we all squared away? We're getting ready to jump into. Yeah. Yep. Okay. So we're gonna do a little concept check here. 2 questions. A photon of which color of light would contain more energy, yellow or purple? Purple. Why? Has a shorter wavelength. Absolutely. Purple, 410 nanometers, the shorter the wavelength, the higher the energy. Contrast heterotroph and autotroph. So basically, what's the difference between a heterotroph and an autotroph? Yeah, absolutely. Heterotroph has to obtain its energy from something else. Autotroph makes its own energy, right? Energy, form of food, right? Sure. All right. Who had me for ninth grade? Tristan, Bella, Val, Scar. Is that it? Alright. So you guys have been this deep into photosynthesis. Even if you weren't in my honors class, right? My basic, if you did not have me, you're gonna go much, much deeper than you ever went before photosynthesis. And that little diagram down there is proof of this. We all good? Ready? Motivate? The light reactions of photosynthesis. Alright, here we go. You need to know this. I'll put it in your little head right now. Here's what we're doing. And right, we've got this in the PowerPoint here, and we've also got this up on the board. What we're starting with is right here. We got a tree, right? Some sort of plant. Water going into the plant, carbon dioxide going into the plant, sunlight going into the plant. Right? That's the basis for everything that we're going to talk about. We look at this plant, this tree, and we focus in on the leaf. And if you focus in on that part of the leaf, you're going to see a bunch of cells with all these little green dots in there. If we look at one of those green dots, we're going to see this. This is called a, what is this?
Yeah.
What is it? It's a chloroplast, absolutely. This chloroplast has stacks of no. Thylakoids called granum, right? So this is a granum because it's a stack of thylakoids, right? We're going to look at one specific thylakoid. That is what we're looking at here.
That's what's in my video. Like,
It is.
all of that.
Right? Because that's how I teach.
Good.
So we're going to look at one specific thylakoid. What you have to keep in mind, ladies and gentlemen, is to appreciate this entire process and appreciate what it means for life on Earth. I'm drawing one section of one thylakoid. Right? There are multiple thylakoids per chloroplast. There are multiple chloroplasts per cell. There are multiple cells per leaf. There are billions of leaves. Everywhere you look out there and it is green, whether it's a blade of grass, a tree, some other plant, whatever, this is happening at it's unfathomable. You can't put a value to it. How many times this is happening per second wherever the sun is shining? I mean, it's all right, it's an uncalculatable number. We have no idea what it is. Right? And this is what drives 100% of the life here on Earth. What we're going to look at in here, in this thylakoid, this is where the light-dependent reactions happen. We have this structure, so we have the thylakoid membrane, which is where the light-dependent reactions actually happen, in the thylakoid membrane. It's important to keep this stuff in mind for when we get to cellular respiration and we ask you guys to contrast them, right? Compare and contrast. You will see that this is exactly opposite of cellular respiration.
Yeah.
Right? We're in the membrane here. That's where our action is going to happen. Not on the inside, right? This is our, so we've got right here, this little box here, which is this here, this is called PS2, photosystem 2. Then we've got protein complexes here that you don't need to know. It's cytochrome and the PR, you don't need to know what they are. Or PQ, I'm sorry. And then we have this other box, photosystem 1. Named photosystem 2 and photosystem 1 because of the order in which they were discovered. Not because this one is second and this one is first. As a matter of fact, it's the other way around. Photosystem 2 is acted on first, and then photosystem 1. So keep in mind, it goes photosystem 2, then photosystem 1. Cool? It's the order that they were discovered, not the order of events. We've got two little special molecules here we call P680. Chlorophyll P680 absorbs light the best at 680 nanometers, which is why we call it P680. Here we have P700. It absorbs light best at 700 nanometers. P700. Cool? Where is, and inside here we have all the water, right? So when you're like, oh, why do plants need water? Where does the water go? Here it is. What do we call the inside of the thylakoid? Yes, say it out loud. The lumen, right? So the water, its intended place is to the lumen inside the thylakoid, and there it hangs out. And then we've got some hydrogen ions in there, right? More hydrogen ions in here than we have out here. What does that mean? Can the hydrogen ions go through the membrane? Can they go through the membrane? Will they just simply diffuse? No. Why? Because they are ions. Ions do not diffuse across the membrane, right? This goes all the way back to the cell structure, right? This is a membrane system. Those hydrogen ions aren't going to come in here. And these hydrogen ions aren't going to leave. They're trapped. But these hydrogen ions, because there's more of them, really want to leave. Right? They're like, yeah, there's more outside. We got to get out. We got to get out. They can't get out. Yes, they can. They can get out one place, right here. ATP synthase. As a hydrogen ion falls through ATP synthase, it will generate an ATP molecule. There's a little turbine system that will generate ATP, meaning there's ADP and phosphates out here, and when that hydrogen comes through, it turns it and it will join a phosphate to an ADP, making an ATP molecule. Well, cool. Well, that's going to happen, and then these are going to balance, and then this is going to shut down. If this shuts down, we have no more ATP. ATP is energy. Without energy, the plant is going to die. We can't have this balance. We cannot let that happen. If that happens, it all ends. So we have to find a way to continue to have a buildup of hydrogen ions inside the lumen. Remember, like equilibrium equals death, right? You said that earlier. We want metabolic reactions happening. They stop, death. How do we do that? And I'm going to draw it up here, and then we're going to go through the lecture. I gotta take off my shirt for this. Yeah, I'm getting warm. Sorry. All right. So we've got our water here, and water is made up of what? It's not a trick question.
Oxygen and
Hydrogen and oxygen, right? We've got H2Os. H2O, 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 right? An enzyme comes in comes in, and remember, enzymes are specific to a substrate. So the water is the substrate, right? And it fits into that enzyme, and the enzyme starts to work on it, breaks it apart. And so the enzyme works on these guys to break them apart. When they break them apart, what do we get? If I break apart water, what do I have? Two hydrogens and an oxygen. Well, that's cool. So you mean I get hydrogens and I get oxygens. The oxygen, how is oxygen found in nature? What do we call oxygen? If you wanted the abbreviation for oxygen, what do we usually call it? No. O2. Why do we call it O2? Because oxygen is a diatomic molecule. It is found as two oxygens. That's why we call it O2, right? So these oxygens, when they break off of the water, don't want to be by themselves. They really want to hang out with another oxygen, right? So they're going to form a bond with another oxygen, and here we go, waste. If you think about the formula for photosynthesis, right? We've got 6 CO2 plus 6 H2O. Sunlight is going to give us C6 H12O6 plus 6 O2. We've already taken care of the water and the oxygen, right? I haven't drawn all the molecules in here, but this is what we're talking about. The water and the oxygen part of photosynthesis is already done. Cool? That gives us hydrogen. When we break this off, is that going to help us? Do we need hydrogen? Kind of a trick question, kind of loaded. Do we need hydrogen? Yes? Or do we need hydrogen ions? We need hydrogen ions. Regular hydrogen isn't going to help us, right? These, when it gets broken, it is really, really, really important that also the electrons get stripped off of those hydrogens, and we keep adding hydrogen ions to the lumen. Why? So that we keep it in balance, so that hydrogens come out here and make ATP. So breaking of water is hugely important. Why? To get hydrogen ions. That's it. Cool? So we get the hydrogen ions. Great. These electrons, what are they going to do? High energy electrons. We can use them for work, right? That's the energy. Yeah, from chemistry, the energy is an association with the electrons in their position, right? We have photosystem 2. Right here, light energy is going to come into photosystem 2. And it's going to hit the chlorophylls here. These are the pigments, right? And it's going to transfer that energy like a little chain reaction. It's going to get to the P680, and there's little electrons up here. And it's going to give that energy to the electron. The electrons are going to move up here, and as they fall in energy, we draw it like this so we show the energy level of the electron. As it falls, it is also going to pull, it's going to use that energy to pull hydrogen ions across the membrane into the lumen. Again, increasing the hydrogen ion numbers here. So that they want to keep flowing out the ATP synthase so we can keep making energy. Right on? So these electrons are going to go to a low energy state. Some electrons are hanging out here. They're going to go to a low energy state. There's electrons in here. This light is going to hit this photosystem, transfer energy, energize these electrons. These are going to come up and take its place, or these are going to go in and take its place, whichever way the system is working right now. Right? These electrons are going to go up here to NADP+. This hydrogen ion can't just sit out here and exist. Its electron is up here. This hydrogen is going to go up here to join the electron and the NADP+, making NADPH. NADPH is an electron carrier. It's carrying the energy from that electron. So that NADPH has the energy from the electrons. It's out here now. We had ATP made out here now. This and this is going to go to the Calvin cycle. This is the light independent reactions. This is where we get the glucose. Cool, that's the first half photosynthesis. Right on. Understanding why things happen gives you an idea of why the formula is what it is. So now we'll kick it back to the lecture, and hopefully with that and the lecture, everything's going to make sense. So if we look at this right here, the overview, water and light go into this thylakoid. What comes out of it? Oxygen, ATP, and ADPH. How does it happen? This. You've got light energy, right? You've got your water here. Your water gets broken into oxygens and hydrogen ions, right? Light energy comes in here, P680, it excites this system, it gets that electron, the electron comes up through here, pulls hydrogens across the membrane, it falls, it regains energy from this light complex, it goes up here, makes, here's our NADP+, we're making our NADPH, right? The electrons come out here to join it. Our hydrogen ions coming through ATP synthase. Here's our hydrogen, it's gonna go over here. There's our ATP. Cool? All right. So, it occurs in the thylakoid membrane. In photosystems, photosystem 2 and photosystem 1, converts a solar energy into chemical energy. Are we good to change slides? So the light reactions. Chemical energy is in two forms, the NADPH and the ATP. That's our chemical energy from the reactions. Cell accomplishes this conversion by light energy photons to excite electrons. Is it fair for me to ask you what parts of there are catabolic or anabolic reactions? Which ones are endothermic, exothermic? Is there any like phosphorylation? There's, right? All these things are fair game. So again, be able to discuss intelligently using the different terms that we've talked about. How is light important to chlorophyll? Chlorophyll absorbs a photon of light. Electron is here to the ground state, it goes to the excited state. That chlorophyll transfers the energy from the sun. So electron is boosted from a ground state to an excited state, which means it has energy. The electrons don't want to live this way. Right? They want to go to their ground state. It's unstable, meaning it's going to react with something. Falls back to the ground state, releases energy as heat. Emits photons as fluorescence isn't real important to us right now. But just telling you what happens when the energy is released. It's not released as sound. Where you go? Plants. Because energy can be released as light, heat, or sound, right? Heat and light and sound. Plants. They did well, she said. Did you guys do no faults again?
Are you guys continuing?
Because like last week you guys had 0 faults.
Yeah, we had no.
Hmm?
We had no.
What?
I mean, we had no fault.
I know 2 weeks ago you had no faults, that's
Yeah,
what I'm
this
saying.
week we're gonna hit 0 too.
This week you were 0-2?
Are you guys gonna keep going?
Yeah.
No, they're quitting. They're just gonna quit now.
This is the last one.
In Vegas.
No, Vegas is in March.
March?
Yeah. I will be here for 2 days.
I may not be here.
Well, I'm gone for a week.
Going back to my home country.
Yeah, for sure. Because this is. Our photosystems. This is where the action happens, like I drew up here, right? Reaction center, complex of proteins, associated with chlorophyll, and electron acceptor. Okay, that electron acceptor is that P680 and P700, depending on which photosystem we're talking about. She took it, I'm bringing it back to you. Jeez, dude, keep your hands on your own stuff. think antenna for the reaction center, sure, whatever, don't worry about it. So photosystem 2, photosystem 1, the two photosystems in the thylakoid membrane. It's the reaction center that holds P680 and photosystem 2. It absorbs light at 680 nanometers. This is all stuff Mr. White already said, huh?
Yes, this is all what Mr. White said.
That's right. Photosystem 1, reaction center, P700, cell absorbs light best at 700 nanometers. Named in the order of their discovery, not in the order of reactions. So these should all be quick notes. Quick diagram.
What's that?
What? I wish I was tech savvy. I would make like a little YouTube channel and stuff along this. I should hire somebody to like follow me around for a year. Or should be someday people are watching YouTube and it just me sitting at my desk, napping,
Yeah.
snacking, things like that.
You'd be like, I could imagine you creating like an office, like the office, like that's how you
Mhm.
act.
Heck yeah. That'd be awesome.
That's great, you should actually.
Are we ready? Everybody good?
28.
Oh, wow, we can finish this.
Bruh. Well.
Alright, so looking into the photosystems, the next few slides, electron flow through the thylakoid membrane will be examined. The picture above will be broken down into steps. We will start at photosystem 2. This is everything I walked you through over here. Right, now we're gonna break it down. Hopefully it is easier to understand.
I mean, if we could understand that,
Right? So, alright, inside photosystem 2, here's our pigment molecules, here's our P680, so these are the chlorophylls. They're gonna be in here, right? Light energy is gonna hit a chlorophyll, it's gonna be transferred to other chlorophylls, it's just gonna keep transferring the energy over and over and over. Causes an electron to go from an excited state back to a ground state. This repeats until it reaches the P680 pair of chlorophyll molecules. So it's going to be ding, 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 ding. We're going to keep transferring that energy and moving the energy along. I'll wait in case you're writing this stuff up on the side.
Mr. White, what do I name the YouTube video?
Photosynthesis. Light dependent reactions. Probably part 1. Or photosynthesis part 1. Because then we can do the rest of this and Calvin's stuff on part 2.
Oh, yeah.
Alright. Got the information. So this is what it is. The electron is transferred to a primary electron acceptor forming P680 plus. Here we go, the electron going up here, right?
Huh?
We're still here. Oh, wait, yeah, here. This is where our water is split, right? Gives us our 2 hydrogen ions because the electrons are coming down here to take the place, right? That's what we've said before. And where this says 1/2 O2, because this is an H2O, I don't want you to be confused. H2O only has one oxygen, right? So we say it's half of an oxygen molecule. So for every one water that's split, we get 2 hydrogen ions, 2 electrons, and half of an oxygen molecule. Cool? Alright, we will pick it back up here tomorrow.